Imagine if we could actually make our old cells act young again. I mean, not just, superficially, like with a good moisturizer, but really deep down, changing their actual genetic programming. Just think about that for a second.
Yeah, it really does sound like something out of science fiction, doesn't it? But that's exactly the promise behind this really exciting area in aging science called cellular reprogramming.
Hmm.
And right at the center of our deep dive today is this one particular gene that's generating a lot of buzz. It's called SB000.
Absolutely. So our mission today for you is to really unpack how this whole idea got started. We want to explore what makes this SB000 gene so captivating right now, and then, really importantly, take a critical look at the big questions, the caveats surrounding its potential. We're pulling our insights from discussions around the SB000 gene, cellular reprogramming, and the quest for youth. So let's get into it.
Okay, so to really understand SB000 neuro, we kind of have to start with the basics, with the foundation, which is cellular reprogramming itself. Now, this whole concept, it isn't brand new. It actually burst onto the scene back in 2006 with some Nobel Prize-winning work.
Right, I remember that. So what exactly did they discover? How did they figure out you could actually sort of rewind a cell's clock?
Well, what they found was by introducing just 4 specific genetic factors, you might have heard of them, OCTO, SOX2, KLF4, and c-MYC.
Ah, the Yamanaka factor.
Exactly, the Yamanaka factors. They found that putting these into adult cells could basically hit a reset button. It wasn't just a small change. It fundamentally turned these adult cells back to what's called a pluripotent state.
Okay, hang on. Pluripotent state. For anyone listening, what exactly does that mean?
Good question. Think of it like a cellular blank slate. A pluripotent cell can become any other type of cell in the body. It's like the super versatile cells you find in a really early embryo. So you could take, say, a skin cell and make it forget it was ever a skin cell, reverting it back to this incredibly adaptable, almost brand new form.
Wow. Okay, that sounds like a massive breakthrough. Revolutionary, even. But it wasn't all smooth sailing, was it? There were some pretty major problems that cropped up almost immediately.
Precisely, yes. While it was, monumental, it hit this huge roadblock. You turn back the clock, fantastic. But the cost, well, first, the cell loses its identity. That skin cell forgets what it is. And worse, much worse, it increased the risk of the cells becoming cancerous, becoming tumor-prone. And that's obviously a massive safety concern if you're even thinking about using this in people. Huge red flag.
Right. Okay, so that danger, that cancer risk, forced to rethink.
Yeah.
The scientific community had to kind of pivot, yeah. And that's where this idea of partial reprogramming comes into play. It sounds like a more subtle approach.
It is, yes. A really critical shift in thinking. Partial reprogramming is about trying to, let's say, nudge cells towards being younger, biologically younger, but without totally erasing their identity. And crucially, without increasing that dangerous cancer risk. You want the youthfulness, essentially, without the terrible side effects.
Makes sense. And this seems to be the area where a UK-based company, a startup called Shift Bioscience, is really making waves. They're kind of leading the charge in this partial reprogramming space, right? Who's behind them?
Yeah, they're definitely one of the key players. The company is led by Dr. Greg Bailey, and their CEO is Dr. Thomas Rando. And Dr. Rando, well, he's a very well-respected name in the broader field of stem cell biology and aging research, so serious scientific background there.
Okay, so they knew about the dangers of the original Yamanaka factors. What was their game plan? How did Shift Bioscience try to find, a safer way?
They took a very systematic route. They didn't just guess. They started by screening, get this, around 1,500 different candidate genes. These were likely picked based on, previous research hinting they might be involved in aging or cell identity. And then they used this really powerful technology called single-cell RNA sequencing.
Ah, okay, so that lets you see what's happening inside individual cells, not just a big mush of them.
Exactly. It gives you super detailed information, cell by cell. So they could meticulously track how each of those 1,500 genes affected the cell's behavior, basically creating this detailed molecular map of what might be pushing cells towards youth.
Okay, 1,500 genes. That's a lot to sift through. Did one particular gene kind of rise to the top, the one that Shift is now focusing on?
That's right. And that brings us squarely to this gene that they've designated SB000. This is the one that Shift Bioscience currently believes might, just might, hold the key to doing this safely rejuvenating cells without the big risks. And the early claims, based on their own work, are pretty tantalizing. Their unpublished data suggests that just introducing SB000 into cells seemed to reverse some key markers of aging.
Whoa, okay, reversed aging markers. That sounds Huge. But how? How are they actually measuring that reversal? What's the yardstick?
Right, good question. They're using their own proprietary tool, a gene expression clock that they've named Agent Clock 3. And this, I think, is where we really need to pause and put our critical thinking caps on, because measuring biological age in a lab, it's complicated. And the validity of different biological clocks is a really hot topic.
Yeah, that feels crucial. Because the established methods, the ones most scientists kind of trust, usually look at something different, don't they? They look at DNA methylation, which is more like stable chemical tags on the DNA itself, almost like wear and tear patterns that build up over years,
Yeah.
relatively stable.
Exactly. Methylation clocks reflect more long-term changes, but Shift's clock, Agent Clock 3, it measures gene expression. Now, gene expression tells you what genes are active right now. It's more like a snapshot of the cell's current activity level. And that activity can be influenced by, well, all sorts of things in the short term, what the cell was fed, stress, you name it. So while it's an interesting approach, this specific gene expression clock they're using, it's not yet widely validated by the broader scientific community. That's a key point.
Okay, so it's their own internal measure. And they're saying that, according to this clock, some markers went backwards by decades. But, and this seems like a big but, these results are purely from cells grown in a lab dish, right? We're not talking about actual living creatures yet.
Absolutely critical distinction. This is all in vitro in glassware. There are absolutely no published results in animals, let alone humans. And perhaps the most important caveat right now, none of this work, none of these claims about SB000 has gone through peer review. That's the standard process in science where other independent experts rigorously check your methods, your data, your conclusions before it gets published in a reputable journal. That hasn't happened yet for SB000.
Right, so lots of excitement, but still very, very early days and needs outside validation. Okay, so stepping back a bit, what does this mean for the bigger picture of aging research? Shift Bioscience, they're not working in a vacuum, are they?
Oh, not at all. This whole field of trying to reprogram cells to fight aging, it's moving incredibly fast. You've got other really high-profile groups pushing the boundaries, too. People like David Sinclair's lab at Harvard, Juan Carlos's Pisua Belmonte, who works in the U.S. and Spain. It's a very active, very competitive research area.
And it's probably important for us and for you listening to remember that aging itself isn't just one single thing you can fix, is it? It's like this whole cascade of different problems happening in our bodies over time. You've got DNA damage building up, mitochondria, the powerhouses getting less efficient, proteins folding wrong, epigenetic changes, the immune system declining. It's complex.
That's a fantastic point. Aging is profoundly multifactorial. It's not like flipping one switch and suddenly you're young again. It involves breakdowns across so many different systems. And what that really suggests is, well, the ultimate solution probably isn't going to be a single silver bullet gene like SB000, even if it proves useful. It's much more likely we'll need a combination of approaches, maybe a silver arsenal, if you like, targeting different aspects of aging. SB000 might become one tool in that arsenal, but probably not the whole solution.
Okay, so let's try and bring this all together for you, this SB000 gene story. It's definitely exciting. It's provocative science. And it might one day, potentially, become part of how we tackle aging. But as we've really dug into, we're just not there yet. Not even close, really.
Yeah, the journey from a finding in a petri dish to something that actually helps patients, it's incredibly long, it's difficult, it's expensive. So many promising discoveries that look amazing in the lab, they don't pan out when tested more rigorously in animals or people. It's a tough road, filled with potential, but also many, many hurdles.
Still, there's reason for some cautious optimism here. Every new discovery, even these early ones around SB000, helps us learn more about the fundamental language of youth that's sort of encoded in our DNA. With every step, maybe we inch just a tiny bit closer to a future where aging isn't this inevitable decline we just accept. Maybe becomes something we can manage, delay, perhaps even someday reverse parts of. So the final thought for you to take away is, what possibilities does this whole scientific journey, this quest to understand and potentially influence aging, what possibilities does that open up for you?